Thank you for the introduction. And today I'll be talking about how deleting tissue B-cell dynamics prevents angiotensin-II induced hypertension in mice and how my work relates to heart failure. So heart failure with preserved ejection fraction, or HFpEF, and heart failure with reduced ejection fraction, or HFrEF, share several risk factors such as obesity, diabetes, and hypertension, with hypertension being one of the significant risk factor contributing towards heart failure progression. Findings from an epidemiological study in the United States, which pooled 4 cohorts across the country with a total of 40,000 participants, recently showed that the absence of hypertension is linked to a lower risk of incident heart failure. It has also been shown that B cells are involved in HFrEF. A lot of studies have shown that their depletion reduces cardiac hypertrophy and fibrosis in mice. Even though the role of B cells have been implicated in HFrEF, their study in HFpEF is limited due to its complex pathophysiology. Recently, in December 2023, in the Journal of Clinical Investigation, a paper was published which showed that B cells in the heart were increased during the first 5 weeks of a two-hit model of high-fat diet and L-NAME. The CD45 positive, B220 positive B cells were increased in the heart, supporting an enhanced inflammatory state of the heart in the early stage of HFpEF. Building on these findings from the recent JCI study, which highlights increased infiltration of the CD45 positive, B220 positive B cells in the heart, my project is to understand the role of B cells in hypertension, and hypertension being a significant risk factor for HFpEF, understanding the role of B cells in hypertensive responses can provide crucial insights into the inflammatory mechanisms driving the heart failure progression. By linking these pathways, my work aims to uncover potential therapeutics for managing both hypertension and heart failure. To study the effects of B-cell depletion, I'm working on an inducible B-cell depletion mouse model called CD19-CRE crossed with ROSA26 IDTR mice. In these mice, the diphtheria toxin receptor has a stop codon upstream of it, flanked by LOX-P sites, driven by ROSA26 widespread gene expression locus. The CRE recombinase here, which deletes the stop codon, is CD19-driven, which is a B-cell specific marker, so whenever we inject the mice with diphtheria toxin, the B cells get ablated. I implanted these mice with angiotensin-II, which induces hypertension, and showed that during hypertension, here shown in blue open squares in the middle, that B cells were significantly depleted as compared to all other controls, here shown blue filled squares, CD19-CRE ROSA26 IDTR mice injected with saline, whereas red here, ROSA26 IDTR heterozygous mice, which have normal CD19 expression, saline injected and DT injected, that the B cells were not depleted. I also showed that during angiotensin-II induction that hypertension is prevented in B-cell depleted mice as compared to all other 3 controls. So deleting tissue B-cell dynamics prevents angiotensin-II induced hypertension in mice. To study the effects of B-cell depletion on HFpEF, I'm working on a 15-week-long model of high-fat diet and L-NAME, but looking at the early stage of the disease. I will use both my experimental and control mice, feed them with high-fat diet and L-NAME, followed by chronic B-cell depletion of 1 injection per week for 5 weeks, and follow them with echocardiography for diastolic function, GTTs for glucose metabolism, flow cytometry to show that B cells were depleted, and body weight measurements. And I showed that here in blue, non-depleted mice had significantly increased body weight as compared to my B-cell depleted mice, here shown in red, as compared to black, that is normal chow-fed mice. On the right here, through global longitudinal strain, I also showed that non-depleted mice have diastolic dysfunction as compared to my B-cell depleted mice. Even though the data is preliminary, we are working on these experiments to increase the N. So B-cell depletion blunts weight gain and diastolic dysfunction in HFpEF mouse model. To summarize, chronic B-cell depletion blunts angiotensin-II-induced hypertension in mice, and also blunts weight gain and diastolic dysfunction in two-hit model of HFpEF. Clinical implications is that an anti-CD20 antibody, rituximab, is commercially used to deplete B cells in autoimmune diseases like rheumatoid arthritis. Will this work in HFpEF is the question that we want to answer.